Hi there, welcome to my channel. I'm Ronald Jenner and this is the very first time I'm doing a digital journal club, so I'm a bit excited. Now, I thought what better way to kick off a series of videos to discuss the evolutionary literature than by giving a little bit of love to a group of creatures that have become quite unloved in recent times. And that group of creatures, of course, are the bats, because of the possible implication as the vector of the COVID virus and transmitting it to humans. Now, this paper was published in April of this year, published in the journal Count Biology. It is titled Embryonic Evidence Uncovers Convergent Origins of Laryngeal Echolocation in Bats. Perfect title, because the conclusion of the paper is right there. The question being, did bats evolve echolocation once, or did they evolve it twice independently? Now, I don't have a pre-existing fascination with bats at all. But this paper I chose as well for a different reason, because it beautifully illustrates, although inadvertently I think, how evidence and the imagination of scientists interacts in a complex way as they draw up their hypotheses and draw conclusions from that data. And so by discussing this paper, we'll tease out exactly what I mean by that. Now, current biology helps the general reader, the non-specialist, by providing a nice graphical abstract, so you can immediately see the two contending hypotheses discussed in the paper, and a few sentences of text in plain English to explain what the authors did and what they have found. And for them, it is clear-cut, it is very cut and dry. The hypothesis of a single origin of echolocation in bats is rejected by their evidence. They say there's two reasons for this. When you look at the development of the inner ear of the bats, which is of course the sensory organ involved in perceiving echolocation, if you look at the development and structure of the inner ear in non-echolocating bats and in non-bats, outgroups, other mammals, it's a very similar picture, almost indistinguishable structure and development. But then secondly, if you look at the development and structure of the inner ear in echolocating bats, they look very different. Not just between the echolocating bats and the non-echolocating bats and mammals, but also between the two lineages of echolocating bats. And for them, that is sufficient reason to, as they phrase it, to suggest that the laryngeal echolocation evolved convergently among bats. So far, so clear. Now let's look at that a little bit detail, whether the evidence presented actually warrants such a conclusion. First, we need to introduce the names of these groups of bats. As you see here in a little phylogenetic tree of the bats, there's three main living lineages. If we start on the right-hand side, one of the echolocating lineages is called the Yancochiropterans. The other echolocating lineage is called rhinolophoids. Rhinolophoids are the horseshoe bats and the leaf-nosed bats. These are bats with really freely ornamented noses. And the third lineages of non-echolocating bats are called the Pteropodidae. And these are the old world fruit bats, quite large bats like flying foxes. I say non-echolocating, it's not strictly true. There is a genus of pteropodids, Rosettas, that contains species that do echolocate, but they don't use their voice box, their larynx, they use tongue clicks to echolocate, but that produces a very functional and sensitive kind of echolocation. It just uses a different bit of anatomy to produce the sounds. Now, if you look at all the data that these authors collected, and there is a lot they did, they looked at hundreds of specimens of bats and outgroups. They looked at dozens of species to figure out how they developed their inner ears. And you can see, for instance, in this figure, this is figure 1 from the paper, this is a blown up version of the tree that is part of the graphical abstract. You can see here along the side view of some skulls of the different bats and the outgroups with in color the orientation and structure of the inner ears. And then they did all kinds of measurements. They measured the width of the skull of bats, the height of the skull of bats, the length of the skull of bats, and they compared those measurements to the cochlear volume. The cochlea, of course, is that spiral bone within which lie the sensory receptors you use for hearing, and bats use to perceive, echolocation. And they then compared the measurements of the skull versus the measurements of the cochlea to see what the relative sizes were. And if you look at this figure, for instance, you can see that if you compare the size of the cochlea and the size of the skull than in outgroups and in non-echolocating bats. This is a small ratio. That means that there's a relatively small cochlea, and in the two echolocating lineages, they have relatively large cochleas. And in this figure, you can see that if you look at the developmental trajectories of the development of the inner ear, you can see that in the non-bat mammals and in the pteropodids, you have the two lower curves. As the animal grows, it gets larger skulls and larger inner ears. But if you compare those measurements with the echolocating bats, you can see that they develop through ontogeny increasingly disproportionately large inner ears. Of course, not surprising, since these are the tools used to echolocate. And then if you look, for instance, also at the timing of developmental events, at the ossification of the bones involved and their hardening, then in the outgroups and in the non-echolocating bats, these bones involved in hearing ossify late, and in the echolocating bats, they ossify earlier. An image like this shows you the three-dimensional morphology of these inner ears. For an invertebrate zoology like me, this is completely impossible to understand, but they provide a useful summary cartoon in which they, with drawings, explain what they already said in the abstract. If you look at the outgroups and the fruit bats, they have almost indistinguishable development of the inner ear. And if you look at the echolocating lineages, you can see that they have a very different development of the inner ear, both from the outgroup and the fruit bats, and also from each other. And that, of course, is the evidence the authors use to say, yes, unambiguously, these things have evolved along diverging pathways, echolocation. So there was also, I just wanted to show this figure to show that there was a lesson to be learned here. You see a summary of the relationships between the species they studied in detail, for each species, 4 developmental stages of the development of the bones involved in echolocation. And even if I blow this up along the entire width of my 21-inch computer monitor, I can make head nor tails of this. This is just too small. It looks nice when you glance at the image. It's like, oh, it's colorful and there's some structure there. But if you want to use this figure to get some information out of it, well, good luck with that. You'd be hard-pressed to do that. Now, if you look and how they actually come to their conclusions, this evidence presented is richly documented, seems compelling to them. But when you then start reading, and you don't have to be a bat expert, the way they conclude that bats evolved echolocation twice is not so much dictated by the evidence, although they present the case, they present it as such. It is dictated by their particular preference for a particular evolutionary story. And one hint of this is already visible in the language they use. So when you look at the 2 echolocating lineages of bats, which are lineages that diverge one after the other, they label them as distantly related, which is interesting because if you would substitute these 3 lineages of bats with humans and bonobos and chimps in the same configuration, I don't think anybody would say that, well, humans and bonobos or humans and chimps are distantly related. They aren't. They are 3 lineages that are each other's closest relatives. They're more closely related to each other than to anything else that exists on Earth. And that is the same with these 3 lineages of bats. So, but by setting up this linguistic flavor, by giving readers the impression that the 2 lineages with echolocation are distantly related, they give you the impression that you perhaps shouldn't believe that anything you find in these lineages has anything to do with each other. So they sort of load the dice already against interpreting what you find as homology in these lineages, but perhaps more likely, as independent developments. And this sort of biased way at looking at things becomes very obvious when you see how they present their alternative hypothesis. So here you can see there's 3 hypotheses they think their data test. The first one is echolocation evolved in the bat ancestor and was lost in the pteropodids. The second hypothesis is hypothesis 2a, echolocation evolved convergently in the 2 lineages that echolocate, the conclusion they prefer and say their evidence supports. But then there's a third one, hypothesis 2b. And that's a weird one, because when I read the hypothesis, like, well, actually, that you already put the hypothesis in. That's hypothesis one. It is completely identical, but they phrase it in a different way, and it allows him to present it as if it is different. So the way they phrase it is that the bat ancestor had some primitive echolocating capabilities, but this was lost in theropodids. And laryngeal echolocation subsequently evolved divergently in Yanko-chiropterans and Yin-pterochiropterans. Yin-pterochiropterans is the grouping of the rhinolophoids and the pteropodids. But hypothesis 2b, when you rephrase it in a more concise way, is identical. It is a single origin of echolocation, and then the diversification of echolocation in the two echolocating lineages, and the loss of echolocation in the pteropodids. It's identical. I don't know how they managed to get this past a peer review, because they are identical, but they phrase it as if these were different hypotheses. And then when you read further, you sort of understand why. They have a very interesting conception about evolutionary change and what you should expect to happen. So I'm going to read this out. When they present hypothesis 1, they say, under the single origin hypothesis, we expect that laryngeal echolocators will share similar development and growth trajectories for the ear apparatus, with minimal differences between the two echolocating lineages. Why? Minimal differences? The species in these two echolocating lineages have diverged many millions of years ago, and evolution doesn't stop when lineages diverge. It's not like you have an escalator that drops off taxa at a distant. Well, let me rephrase this. Evolution is descent with modification, as Darwin phrased it. In modern terms, it's descent with infinite, potentially infinite divergence and diversification. So evolution continues after lineages split, because if it didn't, we would probably still see life only as some sort of bacterial froth that never got anywhere. So evolution will not be restricted. You will not have an expectation of minimal evolution after divergence of lineages. That's a very weird, at best an old-fashioned way of thinking about evolution. And then if you look at how they phrase, well, the hypothesis that if there was a common origin of echolocation, theropodids must have lost echolocation. Is that likely? And they say, well, no. Again, their expectation is that embryonic development, especially early, is highly conservative, and completely erasing the signatures of ancestral development in descendants is unlikely to happen, even if the morphology expressed by adults is highly modified. Well, maybe, but evolution has countless examples of morphologies being reduced and completely lost as well. What about those? It is certainly not impossible a priori. Well, they say, no, look at mice. Mice in evolution 50 million years ago or so lost their premolars, but you can still see vestiges during development. Okay, so see, it's not erasable, that history. Okay, and so, well, look at dolphin development and baleen whale development. Dolphins still have vestiges of hind limbs in embryos. Okay, and baleen whale embryos still have vestiges of teeth. So, you know, it's almost impossible to erase these ancestral traces of previous morphological structures. So, right, we don't see these in the pteropodids in the fruit bat. So, well, there's no evidence they lost any of the morphological specializations to do with echolocation. So probably they didn't evolve from an ancestor with echolocation. But they included a genus, a species in the genus Rosettus, in their analysis. They looked at its inner ear. It develops pretty much indistinguishable from the outgroups, the non-bats, but this species echolocates. It uses tongue clicks to produce the sound, but apparently with this very minimally, if at all, modified inner ear, they can still functionally echolocate quite well. So the hypothesis that the common bat ancestor did have a form of echolocation, which was then lost in fruit bats, and continued to evolve and diverge in the two echolocating lineages, is not impossible. In fact, when you look at the evidence presented, it's completely and utterly consistent with everything presented there. but the authors still prefer to go for the convergent evolution of echolocation hypothesis. And then it gets really interesting, because if you look at their figure 2D, they actually did a formal phylogenetically structured comparison of the ontogenetic trajectories of the inner ear development in the different species. And they did an ancestral state reconstruction to ask the question, did or did not the bad ancestor likely have echolocation or not? And when you look in this phylogenetic tree, and you look at the little node that is numbered 2, that is the last common ancestor of all bats, then their own quantitative phylogenetic ancestral state reconstruction suggests that it is likely, and they even give it a number, 66% posterior probability, that the bad ancestor did in fact have laryngeal echolocation that was subsequently lost in fruit bats and maintained in the 2 other lineages. So there you have it. The paper was published based upon preferring a certain set of evolutionary intuitions, gut feelings about what the authors thought was likely and not likely in evolution, in actual conflict with their own quantitative phylogenetic analysis. And that is fine. Evolutionary reconstruction is a complex task. The peer reviewers allowed them to do this, which I found a little bit puzzling, because nowhere in the paper do they really grapple with the question, well, could these differences of inner ear development that you see in the echolocating lineages, could those be due to descent with modification rather than convergent evolution? They pay lip service to it, but they don't really properly assess it. And in my view, I hasten to add again, I'm not a bad expert, the result they presented is perfectly compatible with the hypothesis for the shared common ancestry of echolocation in bats. And perhaps that bad ancestor had an inner ear structure quite similar to some of the echolocating fruit bats living today, a minimally modified form. Now, that's an interesting story, I thought. I've just finished writing a book called Ancestors in Evolutionary Biology, Linear Thinking About Branching Trees, and the main red thread, golden thread, I think I'm Dutch, we say rode draad, which is the, I think in English you say golden thread that runs through a story. Anyway, the golden thread that runs through the story of my book is how the preconceptions, gut feelings, background knowledge, often from ill-defined sources that individual researchers have, it often colors, sometimes even dictates the conclusions they draw from sometimes enormous amounts of empirical information. And this paper is a beautiful illustration of this, where the evidence is clearly open to alternative interpretations, which I do admit the authors do give that some discussion, but the conclusions they then unambiguously present and prefer are actually contradicted by their own quantitative phylogenetic analysis. And that is an interesting finding, I thought. Now, this is sort of elephant in the room. Can you actually use observed differences in the structure of things or the development of things as evidence to reject common origins or not? And that is the broader context with which you can discuss this paper as well. But I've already spent almost 20 minutes here. That's long enough. Then we'll have to wait for another video at some other later point in time. In closing, I do want to say, when you read the acknowledgements of the paper, they make a point of thanking a late colleague of theirs, Tomonari Kaji, a very young, but very talented researcher, an evolutionary morphologist who sadly died in his 30s, and they dedicate that paper to his memory. He wrote, and I'm familiar with some of his work, he wrote some extraordinarily cool papers on the evolution of morphology of invertebrates. For instance, how termite soldiers evolved these glue guns on their heads, or how snapping shrimp evolved super fast closing claws with which they can stun enemies or food, and a few other papers. So the Tomonari Kaji's papers are really worth reading if you are interested in the evolution of morphology and the emergence of novel features. Now, if you like this, if you thought this was distracting you from doing your work on the cross trainer or eating your lunch, please like it, subscribe, and sign up for alerts, so you get a warning when I have another bit of evolutionary literature to riff on. For now, it's getting really nice and sunny. It's Friday afternoon for me. Have a good weekend and hopefully see you next time. Bye bye.